Moving to the topic sex-linked inheritance. Sex is determined by the presence of a pair of dissimilar chromosomes in one of the sexes. For example, in both humans and Drosophila, the male consists of a pair of X and Y chromosomes, whereas female consists of a pair of two X chromosomes. The Y chromosome is smaller when compared to the X chromosome. It is relatively inert and has fewer genes when compared to the X chromosome. Thus, the genes present on the X chromosome exhibit unique patterns of inheritance. The term X-linkage is used to describe such situations. This inheritance pattern, due to presence of certain genes on X chromosome and their absence on the Y chromosome, leads to modification of the Mendelian ratios. So first we'll see X-linkage in Drosophila. H.Morgan first documented X-linkage in the year 1910 while he was studying Y-type mutation in the fly Drosophila melanogaster. Normally, the Y-type Drosophila has red eye color, which is dominant to the white color of the eye. Now Morgan studied the F1 and F2 results of reciprocal crosses. Reciprocal crosses between Y-type and mutant flies did not follow the Mendelian ratios. That is, when the sex of the parent showing Y-type trait was interchanged, the results were different. It was concluded that the white locus, or the gene associated with white eye color, was located on the X chromosome, and thus both the gene and the trait are said to be X-linked. Here we have the reciprocal crosses studied by Morgan. In cross A red-eyed female is crossed with a white-eyed male, while in cross B, a white-eyed female is crossed with red-eyed male. In F1 generation, half of the progeny was found to be red-eyed female, while the other half was red-eyed male offspring. While in the case of cross B, half of the progeny was found to be red-eyed female and the other half was white-eyed male offspring. When this F1 generation is self-fertilized, F2 generation is obtained, where in the case of cross A, half of the progeny was red-eyed female, 1/4 was red-eyed male, and 1/4 was white-eyed male offspring. Whereas in the case of cross B, in F2 generation, 1/4 progeny is red-eyed female, 1/4 is white-eyed female, 1/4 is red-eyed male, and 1/4 is white-eyed male offspring. So here we have the X and Y chromosome of a male Drosophila. On the X chromosome is present the locus or gene responsible for the eye color. Its corresponding position on the Y chromosome is absent, as the Y chromosome found in males is much smaller. Whereas the females have two gene loci available, one on each of the X chromosome. As the Y chromosome lacks homology with most of the genes on the X chromosome, whichever alleles are present on the male X are directly expressed in the phenotype. And because the males cannot be homozygous or heterozygous for X-linked genes, this condition is known as hemizygous. In such cases, as no alternative alleles are present, the concept of dominant and recessive is irrelevant. Let's see the chromosomal explanation of the reciprocal crosses observed by Morgan. Here we have a female Drosophila showing two X chromosomes, which is homozygous dominant for red eye character. The dominant gene for red eye color is represented here by capital R. This female Drosophila is crossed with a white male having a recessive gene small r on the X chromosome and a Y chromosome. The following types of gametes are produced, and in the F1 generation, the following combinations of progeny is achieved. In the F1 generation, we have a heterozygous red female and a red male. This F1 generation is now self-fertilized, and following types of gametes are produced. In the F2 generation, the following combination of progeny is achieved. We have a homozygous dominant red female, a red male, a heterozygous red female, and a white male. Similarly, in cross B, we have a homozygous recessive white female and a red male. The recessive gene is represented by small r and the dominant red gene by capital R. The following types of gametes are produced, which result in a heterozygous red female and a white male. Now this F1 generation is self-fertilized. The female produces the following gametes and the male produces these gametes. Using a Punnett square, the results of F2 generation can be known. In the F2 generation, we have the following progeny. We have a homozygous recessive white female, a heterozygous red female, a white male with small r recessive gene, and a red male with capital R dominant gene. Let's move to X-linkage in humans. In humans, many genes and traits are X-linked. These X-linked traits can be observed in pedigrees and are characterized by criss-cross pattern of inheritance. Let's see a few examples of sex-linked inheritance. Color blindness, hemophilia A, where there is a deficiency of blood clotting factor 8. Then hemophilia B, where there is a deficiency of blood clotting factor 9. Muscular dystrophy, where there is a deficiency of protein dystrophin. Then Hunter syndrome, which is a mucopolysaccharide storage disease, where there is deficiency of enzyme iduronate sulfatase.